Now, for our discussion or our review, we will start first with the last three topics, which would be biomolecules, lipids, and also carbohydrates. This would be easier to remember because we just had this for the past few meetings. And starting with biomolecules, you will remember our discussion about the Lego. So, Lego pieces have been used or is used to make very large and nice sculptures like this one, which is the Iron Man. And you know that these large sculptures are made up of the smaller pieces of Legos. And for the Lego, there are only two parts, which would be the stud and also the holes underneath of each of the Lego. So this is the same with biomolecules because despite the diversity of species, all of life on Earth are built with the same structural molecules. There are only a few structural molecules that will make up everything. The whales, the flatworms, the bacteria, and even us. So we are made, or all of the living things are made up of very small molecules, which we call as biomolecules, and only a few of them are used to make up all of life. What are the four major classes of biomolecules? You are very familiar with this one, and let's just try to remember that. Don't forget, it is CLPN, CLPN, and the four major classes of biomolecules are carbohydrates, lipids, proteins, and nucleic acids. So very familiar with us. When we talk about carbohydrates, of course, this would be bread, the starch that we can find in the potato, and several others. Lipids would include uh, cooking oil, different kinds of waxes, and like palm oil and cooking oil. So all of this would be lipids. Proteins, you're very familiar. So we have chicken, majority of which would be proteins. We have the meat of fish. We have pork, so those would be included as proteins, or majority of which would be proteins. And nucleic acids, of course, would be made up of DNA and RNA that we can find inside of the nucleus of all of the cells, or many of the cells. So some things to note about biomolecules. First of all, biomolecules, these are structures that biological macromolecules play a role in their function in living systems. So just remember that the structure dictates the functions, or the function would depend on the particular structure. And the biomolecules are actually made of carbon, or starts with carbon, and it would be a chain of this carbon which form the skeleton of most of the organic molecules. So these carbon chains may vary in length. They may be straight, may be branched, may be close rings. Some may have double bonds, some do not have double bonds. And if they do have double bonds, the location of the double bonds may vary. So this will be showing us that there are indeed a variety of molecules, biomolecules, and the differences in their structure will also give us a variety of functions as well. So another question from our LCG in biomolecules: what do you call molecules with the same number of atoms but have different structures? You already know this. The answer to this is, again, it's letter I, isomers. So as you can see here, letter D, letter C, and letter B, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, and then if you also take note of the number of oxygen and hydrogen atoms, carbon, they will have the same number. The only difference you can see here is that they have differences in their structure. Just like in the letter D, the double bond O is at the end of the carbon chain, and then they they have differences in the position of their OH. For example, for this one, it's on the right side on the second carbon, while the other one, letter C, the OH is on the left side of the second carbon. And then the double bond O would be found in the second carbon for letter B, whereas letter C and letter D is at the end of the carbon. So you will see here that they have different structures, and this will lead to differences in their functions as well. Okay, another question: what affects the properties of these molecules? You know this. The answer to this is functional groups. And of course, you have to remember or be familiar with the different kinds of functional groups and their structure and some examples. So let's just try to remember these functional groups. If you're given OH, the structural formula is OH, and the ball and stick model will look like this. The name of this group of functional, or the name of this functional group is hydroxyl. Just remember H, and O is give us an idea of hydro, so hydroxyl, and this can be found in alcohols like ethanol. Now the other functional group will be COOH, wherein there's a carbon, a double bond O, and an OH attached to the carbon, as you can see also in the ball and stick method. What do you call this particular group? Here's your clue. There's a C over here. So this is carboxyl. And you can find this in acids like acetic acid, like vinegar. So this would be the carboxyl. So we're done with hydroxyl, we're done with carboxyl. How about another one, which is NH2? So this is a nitrogen wherein two hydrogen are connected to the nitrogen. What do you call this particular functional group? You call this as amino. Now take note that an example of this would be bases like ammonia, and another functional group would be SH. As you can see in the structural formula, the sulfur hydrogen is attached or is bonded with sulfur here. And what is your clue here? Of course, there will be an S. The name of this group is sulfhydryl, while this is found in rubber. Last two sets of functional group would be PO4, wherein the 4 of the oxygen would be covalently bonded to phosphate, and one of the oxygen will be a double bond. So what do you call this group? You call this, of course, you have a clue. It's letter P. It would be phosphate, and you can find this in the energy currency of the cell. You can find that in ATP. And then you have here finally the COH and the CO, where the COH would be a double bond O connected or bonded to carbon, and then a hydrogen is bonded to it as well, as you can see here in the ball stick model. And then the other one would be carbon and double bond O. So this is COH and COO would be called as, your clue here is the letter C in front. It will be carbonyl. So don't get confused with the carbonyl and the carboxyl. So the COH is called an aldehyde, while the C double bond O is a ketone. So we'll be talking about that later in the sugars as well. So those are the functional groups that you have to remember: carbonyl, carboxyl, phosphate. You have to remember the hydroxyl, sulfhydryl, and all of those functional groups. So just remember, the addition of these functional groups will definitely change the properties and definitely change the function of those particular biomolecules. Okay, another thing we talked about would be polymer. So what is a polymer? Polymer would be long molecules composed of a number of many identical or similar building blocks. So just remember, putting together different monomers of sugars can make polysaccharides. Putting together many many units or monomers would make a proteins, so it should also be a polymer as well. So just remember, polymer is a long molecule made up of repeating units. Which among the biomolecules are polymers? You have to remember 3. The first one is carbohydrates, another is protein, and the other one is nucleic acid. I think there was a mistake in the PowerPoint in some of our classes, wherein instead of nucleic acid, lipids was written down. So please do remember a correction here, that the biomolecules considered as polymers will be carbohydrates, proteins, and nucleic acid instead of the lipids. So lipids are not considered as a polymer because there is actually not much repeating units there. They're simply made up of glycerol and fatty acids, at least for the simple lipids. So there's not much of a repeating unit as compared to carbohydrates, proteins, and nucleic acids. So next question is, how are polymers formed? We already talked about that last time. So the process in which the polymers are formed are called the dehydration reaction. So in the dehydration reaction, the result is you form a covalent bond between the two monomers, putting them together. And then another important thing is that there is a loss, and this is accompanied by the loss of water, and it is facilitated by enzymes. So polymers are made through dehydration reaction, and this is the example of that. This is monomer 1 and this is monomer 2, and you have an OH and an OH as well on the other monomer, and when there is a reaction facilitated by the enzymes, the OH of one of the monomer and the H of the other monomer will combine together or will be released, so that you will form water, and then there will be the formation of covalent bond between monomer 1 and monomer 2. So you call that as the dehydration synthesis. So where water is the byproduct of this dehydration process. Okay, finally let's look at the classes of biomolecules. So this is very important for you to remember. What will be the building block? What will be the monomer if they are polymers? Let's start off with carbohydrates. So do you remember what will be the building block of carbohydrates or their monomer? It is called monosaccharides. For proteins, the building block would be amino acids. Lipids, they are not considered as polymers, so they don't have monomers, but they do have building blocks, and the building blocks of lipids would be glycerol and fatty acids. While the nucleic acids, what would be the building block or the monomer? It's called the nucleotides. So let's now talk about where in the cell can you find an abundance of this particular biomolecule. So you have here which part or organelle. For the carbohydrates, you can find them in lysosomes, cell membrane, there will be carbohydrates there, and chloroplasts and vacuoles as well. Okay, how about proteins? Where can you find this in the cell? You can find the proteins in ribosomes, the rough ER, and the cell membrane. So even the cell membrane will be containing proteins as well, and an abundance will be found in the ribosomes because ribosomes are the producers of the proteins, and the rough ER, this would be the endoplasmic reticulum that has a lot of ribosomes connected to it, attached to it. That's why there will be a lot of proteins there as well. How about the lipids? Or the organelle or the cell part where you can find an abundance of lipid would be endomembrane system, and don't forget the cell membrane as well. Particularly in the endomembrane, it would be the smooth ER, because a smooth ER produces lipids. And finally, nucleic acids. Where can you find this? Which part of the cell? You know this already. You can find this in the nucleus, but also in the mitochondria and chloroplast. The reason being that we discussed this in Bio 1, wherein mitochondria and chloroplasts are hypothesized to have been derived or evolved from bacteria. So bacteria have their own DNA. So mitochondria has DNA, and chloroplasts will also have their own DNA, thereby having an abundance of nucleic acids. Now for the functional groups, carbohydrates will have carbonyl and hydroxyl. The proteins will have what kind of functional groups? Amino and carboxyl. The lipids will contain which functional groups? Carbonyl, hydroxyl, and phosphate, while the nucleic acid will contain amide, hydroxyl, and phosphate. So this would be the discussion for the biomolecules.